Hey everyone, in this lecture video I'm going to talk about the third topic in regulation of gene expression, which is long-term patterns in gene expression, with a particular focus on expression patterns that occur over the course of embryonic development. And just a small note of warning, I'm going to move through this fairly quickly in this video, since this is a topic that I also talked about during our lecture time. I'm mostly providing this to help anyone who had trouble following along during lecture be able to review. So you may find that you need to pause the video to take notes throughout it, just to make sure that you understand the concepts that are being discussed. So if it feels like it's moving pretty fast, that's because it is. And so what that means is pace things at a pace that works for you. Okay, so I started out talking about some of the traditional study systems for animal developmental biology. So a lot of these organisms are animals that have fairly large eggs that are fairly simple to study directly. So organisms such as frogs or birds, also a small fish called zebrafish, fruit flies of course because they show up everywhere, and in addition to that there's also a type of small worm, a nematode worm called C. elegans that is also used for a lot of developmental biology studies. One of the reasons I bring these up is that we have a couple of professors here at Siena who work with some of these study systems to study different aspects of animal development. So if you find this topic really intriguing, it's a good idea to look for courses that are taught by these professors, or you might want to get in touch with them to find out about their research programs. And although I'm just going to be talking about kind of giving some background on developmental biology, in the modern genomic era, we're now able to study a lot of these questions in a much wider range of organisms. And so in general, across developmental biology, the overall interest is in kind of common features to developmental processes across organisms, so processes that are really shared. And this has really blossomed into a new field in about the past 20 years within biology, the field of evolutionary developmental biology. So if you find this really fascinating, just know that there's a lot of possibilities to continue studying in the subject area. So for thinking about gene expression across development, we have to set the stage by thinking about how development works for a multicellular organism. So, we start out with an egg hitting a sperm, and that brings us back up to a diploid organism. And when it is just a single cell, it's referred to as a zygote. And so the process we're talking about here is a process of going from just one cell to many different cells, and also many different types of cells. And if you think about a multicellular organism such as yourself or myself, or as shown here, a frog, all the cells in our bodies have a wide range of different structures and functions. And over the course of development, a hierarchical organization also arises. So we go from this one cell state to suddenly having different cell types and different tissues and different organs and different organ systems and entire organisms. And that gets worked out over the course of an organism's development. And it isn't just the type of cell that gets sorted out among all of the cells communicating with each other, but also how those cells are arranged in space. There has to be some three-dimensional structural arrangement that takes place as well. So developmental biologists divide up early components of the development process into kind of three major stages. First stage is called early differentiation, and this is a stage where cells that are initially totipotent then differentiate. I'll talk a little more about that in a minute. And this is also linked to the start of specific gene cascades in specific tissues. And again, I'll talk about that more in a moment. Another component to development is called pattern formation. And this is the process of different development zones starting to appear in an embryo. So an identifiable zone that's going to develop into something like legs, the head, or the abdomen. And the genes that control the process of pattern formation are referred to as a collection of genes called the homeotic genes. So that's the second component of development. The third component of development is called axis establishment, and that's kind of that spatial element that I talked about. So this involves the expression of genes that help to indicate on an organism what's going to be the front and what's going to be the back. What'll be the top or the bottom, what's going to be left and right. And the genes that determine this are called morphogen genes. So walking through this process of differentiation, at the zygote stage, so when an organism consists of just one cell, there are intracellular signaling processes that take place. So that's signaling processes that happen within a single cell. And the way we know this is actually through transplantation experiments, where if you slurp the cytoplasm out of one place in a cell and you put that cytoplasm somewhere else, you wind up sending that cell on a different developmental pathway than would happen if you didn't move around some of those elements in the cytoplasm. So that's, at the single cell level, everything has to be in that cell, so it's just based on spatial organization inside of the cell. And then later on in development, usually when an embryo reaches around the 32 cell stage, then there starts to be more coordination between different cells, and so exchange of signals that are called inductive signals. So this is cells talking to each other, saying, hey, I'm going to turn into the head. Oh, okay, I'm going to turn into the leg. So that you don't wind up with two heads and one leg. Instead, one head, two legs. So that's sort of how things get started in these early stages. So again, I mentioned the word totipotency. The early cells, right after the zygote stage, up to about 32 cells, those initial cells are all this word totipotent. And what that means is those cells have the potential to develop into any type of cell in your body. So you could take those cells and they turn into liver cells, it could turn into muscle cells, all kinds of things. And so when you hear about identical twins are groups of zygote cells before they reach this 32 cell stage that each are totipotent. So sometimes they get separated from each other and there's no problem there. The remaining group of cells in each of those subgroups can continue to divide and the signaling processes can take place and you can wind up with twins or even more than twins. So again, so these cells are totipotent. So that means they could be, the potential is infinite, basically, is what this is saying. They could become any type of cell. And that's a big part of why biologists tend to make a pretty big fuss over human embryonic stem cells. Those are the cells that are totipotent because those can allow you to do a lot of different things. For one thing, it could let you grow any different cell type you like. And so that could be really useful, for example, for helping people replace a lost limb or something of that nature. They're also incredibly valuable for just studying the process of development. So figuring out how it is that these early cells go from being totipotent to then having a specific cell fate. We do have some types of cells in our body that retain some ability to differentiate. We refer to those as other types of stem cells, but those stem cells tend to be very specific to differentiating to very specific types of other cells. So, for example, there are bone marrow stem cells that can produce red blood cells. And so those stem cells have some capability, but not nearly the same type of capability as these early embryonic precursor cells. So the process of differentiation involves that cell-cell signaling that causes tissue-specific genes to turn on. And as that happens, that process is usually irreversible. So you go from being totipotent to having a specific cell fate. And what will happen is one gene might turn on, and so what that means is that gene's getting transcribed and translated, so that gene is being expressed, and often the product of that gene also winds up being a transcription factor that can wind up either inhibiting or stimulating transcription and translation of other genes as well. And so this process of one gene then going on to become a transcription factor for another gene, and even more than one gene, that process is referred to as a transcription cascade. So there's an example of this type of cascade in the image on this slide. So this is examining a muscle cell. And so early on in the precursor cell, the cell that's eventually going to become a muscle cell, there might be some kind of master regulatory gene. So here's a gene called MyoD that gets turned on. So the cell learns, okay, surrounding cells are going to become bone and skin, so I guess I'm going to be muscle. So this gets turned on, that transcription factor starts to be produced, and that can do a couple of different things. So this is actually showing that it's enhancing production of more MyoD, and then it's also enhancing production of some other transcription factor here. We don't know what it's called. And that other transcription factor is going to turn on a whole bunch of additional genes that are associated with becoming a muscle cell. So that's an example of one of these developmental cascades. And so when you know that idea, you can kind of take that idea and run with it and think, okay, across different cells, this is going to be happening during development. And that's how we go from this initial totipotent cell to a differentiated multicellular organism. So I want to talk a little bit more about these pattern formation genes. So these are genes that are involved in the establishment of structured growth zones. So the way these were discovered is kind of cool. Back in the 1940s, when geneticists were just bombarding flies with X-rays to create mutations, they discovered a category of mutations that were very strange body structure mutations. So I've illustrated one of them on this slide, and if my head's hiding it, you should look at the lecture slides as well. But this is a mutation in flies that causes a fly to develop legs where ordinarily it would develop antennae. And the coolest thing about these is that these mutations are heritable. So you can map these mutations to specific locations or loci on chromosomes. So what that tells us is this tendency to grow legs out of your antenna holes is heritable, and it must be controlled by specific genes. And so these are the genes, again, that are called the homeotic genes. So they're involved in structural control during development. And we can identify the homeotic genes because there are very specific expression patterns for those genes in particular regions of a developing embryo. So these genes show up, and they basically are the first signal that says this area is going to wind up being where the eyes are, or this area is going to wind up being where the back part of the abdomen is. So this picture that I've included in the gene expression lectures show a set of homeotic genes for a fruit fly. So when these get expressed in the embryo, then they go on to determine where those segments show up later on in the fly. And so you could do something very similar to what I showed with the mutations, where, for example, if you transfer one of these sections expressing the homeotic genes for a leg and you move that somewhere else in the fly, you can then get that fly to grow a leg, like say maybe out of the back of its abdomen or something like that. So that's all regulated by these homeotic genes. One of the other weird mutations that popped up was something that geneticists discovered in the embryo of flies whose mothers are homozygous for a particular gene called the bicoid gene. And so when a fly's mom has a mutation for this, and she's homozygous recessive for this mutant gene, then that fly embryo doesn't develop properly. So instead of developing a head and a tail, what winds up happening is it winds up having two tails. But you'll notice that it still has its body segments in spite of having two tails. So, then geneticists really wanted to know, okay, well, how does this work? So one step for getting there is to come up with some stains that will let you label and locate where the messenger RNA is for this gene product and where that protein is to see where it is present inside of a developing embryo. So if you do that for the bicoid gene and you stain for messenger RNA, what you discover is you can find the messenger RNA actually in an unfertilized fly egg. And even more than that, it's only found in one specific spot in that unfertilized fly egg. It's in one end. So before this egg is fertilized, it's got messenger RNA for the bicoid protein. And so then, if you track things later on, after the egg has been fertilized, then you can find bicoid protein that is also localized in the same location. And that location then develops into the head of the fly. So this leads to a question, which is, well, where's this mRNA coming from that's in this unfertilized egg? And that mRNA actually comes from that egg's mother. So this is a gene that we also refer to as a maternal effect gene, because it doesn't matter what the fly's genotype is. What matters is what its mother's genotype is. That's what determines whether or not this fly winds up with a head and a tail, or whether it winds up with two tails. So bicoid is an example of a morphogen gene that's involved in this process of axis establishment, figuring out head from tail. So that other category of genes that gets expressed during development. Okay, so that's it for the regulation of gene expression. So we've looked at short-term methods in prokaryotes. We've looked at a handful of examples in eukaryotes as well. And hopefully this has given you an appreciation for the complex ballet that takes place over the course of embryonic development. So that you go from that single zygote to a multicellular organism that has all the right organs in all the right places.